All right. So we're going to do peripheral nervous system over a couple of lectures because there's a lot of different ways to look at it. We're going to start with looking at the anatomy of it, and then some of this we're going to kind of touch on again as we get into like the actual physiology of it and how we divide the peripheral nervous system into sensory and motor components, as well as somatic and autonomic components, because there's a lot of overlap there. So to start, we want to look at the anatomy of a nerve. So remember that nerves are just what we call white matter in the peripheral nervous system, which means they're going to be primarily composed of axons and then Schwann cells, which are going to myelinate some of the axons. And then, of course, there's going to be a bunch of supporting materials, right? Connective tissue, blood vessels, for the most part. And then ganglia, or a ganglion, that's singular, is what we call gray matter in the peripheral nervous system. And so that's going to mostly be the cell bodies of neurons, almost exclusively sensory neurons, and then the satellite cells that support them. So when we look at nerves, we see that we can divide them up into two groups, which we'll talk about later. So cranial nerves are all the ones that originate from the brain, pretty much mostly the brain stem. And then spinal nerves are the ones coming out of the spinal cord. So we'll talk about the specifics of them later. For now, we're just going to look at the generalized anatomy of all nerves. So when we look at the generalized anatomy, we see that it's a whole bunch of axons running together, so they're running in parallel, and they're protected by multiple layers of connective tissue, which is very, very similar to what our muscles are organized as. Some of the axons are myelinated, some of them are unmyelinated, and because they're sending action potentials, they're going to be using a lot of ATP because of the electrical activity that we've talked about before. So there has to be a lot of blood vessels in there too, providing the oxygen and the fuel to, you know, give them ATP. There are generally a little bit of mitochondria running with these axons, or they're other places in the cell. Come on. All right, so 3 layers of connective tissue, and they pretty much parallel the layers in the muscle. So all we're doing is we're switching out mesium for muscle to neurium for nerve. Okay, so this is from inside to out. I don't know why I did it that way, but whatever. So each axon is covered with an endoneurium. This includes Schwann cells. So Schwann cells that are myelinating the axons get covered as well. So this is going on the outside of both the axons and the Schwann cells. And then the axons are actually divided into fascicles within the nerve. And so each fascicle is bundled by a connective tissue perineurium. And the largest blood vessels inside the nerve are going to run in the perineurium in between the fascicles, and then branch into tiny blood vessels to each axon. And then the entire nerve is going to be bundled in an epineurium, a nice dense connective tissue that keeps the whole thing together and helps protect all the axons from external damage. So we can look at a diagram like this and see all of those things. So epineurium surrounding the entire nerve, and then we have these bundles or fascicles of axons. You can see blood vessels running in between them. And then some of them are unmyelinated. And you can see there's still Schwann cells associated with them, but they kind of wrap around a bunch of them and just hold them together, versus the myelinated, where we have actual nodes of myelination. And this doesn't really show you the endoneurium super well. It's got endoneurial fascicle around some unmyelinated fibers here. Here's the one in your textbook, showing basically the same thing. And I kind of like this one because it gives the fascicles a little bit of a better organization. But yeah, so each individual axon with a perinerve around it. So when we talk about nerves, this is what we're talking about. And then when we're talking about actual pathways and a neuron, we're talking about the individual axons that are running in the nerves. Here's another of how we are pretty sure the Schwann cells hold the unmyelinated fibers. So even though Schwann cells' primary job is to myelinate in the peripheral nervous system, they also just generally hold all axons so that they're not getting like tangled up in the nerves. So this is how they hold them when they're not myelinating them. They can hold a bunch of them. We find that there's different ways to classify nerves based on the direction of information flowing in the axons. Now, the vast majority of nerves are mixed in that they're carrying sensory axons back to the central nervous system and motor axons from the central nervous system. All spinal nerves are like this. And I'm not going to spend a lot of time dwelling on this particular concept because almost all nerves are mixed. But when we get to the cranial nerves, we'll see that it's possible to have a purely sensory nerve, so one that's only sending sensory information back to the CNS. So we call these sensory or afferent nerves. And we can have purely motor nerves as well, or efferent nerves, where they're only carrying information from the CNS to effectors. Axons within nerves, and occasionally nerves if they branch enough, are classified as being either visceral, and remember visceral is basically synonymous with somatic, or autonomic and visceral are the same thing, or somatic. And we're going to talk way more about that later, okay? So we're not going to get too hung up on this classification, but I just want you to be aware of it because when we look at cranial nerves, they part of the way that they are described is by whether they're mixed sensory or motor, okay? But all spinal nerves are mixed, and it's kind of silly to dwell on it. And then this is just a microscopic picture of a nerve, and hopefully if you haven't yet, you'll look at the histology slides that we're doing, and this will start to look more familiar, but you know, you can see those layers of connective tissue when we look at these under the microscope too. So then the ganglion, as I said, are a bunch of cell bodies. And there's two locations that we see gray matter in the peripheral nervous system. So the dorsal root ganglia, which I don't remember if I mentioned when I went over the spinal cord, are located in the dorsal root of the spinal nerves as they come off of the spinal cord. So with the spinal cord, we have sensory information running dorsally, motor information running ventrally, and all that sensory information, the cell bodies of those fibers are actually here in these dorsal roots. So dorsal root ganglia are always sensory neurons. And then there are also autonomic ganglia, which contain the cell bodies of autonomic motor neurons. So these are always motor. And a lot of them are located right here. And we'll talk more about these guys. This is called the sympathetic chain. We'll talk more about that when we get to the autonomic nervous system. So here's a picture of that. And this is when we see literally neurons in a peripheral structure. And as usual, neuron cell bodies, if you guys haven't noticed this yet, they're huge. compared to other types of cells. And so then all these little dots here are the nuclei of the satellite cells surrounding each cell body. So there's a lot of satellite cells to cover a neuron because the neuron cell bodies are so much bigger. So these are the pictures again that are in your textbook. All right. So the big thing that we like to focus on when we talk about the peripheral nervous system is the sensory input and the motor output. Because remember, almost all, all of the integrative functions are by the CNS, right? So the central nervous system, the brain and the spinal cord, they do all of that second function of the nervous system. But the first function, the sensory input, and the third one, the motor output, that's all peripheral. So we're going to start with looking at some of the sensory stuff, and then we're going to look a little bit at the motor output, but then we're going to go into more detail about it as we talk about the somatic versus autonomic, okay? So this is an introduction to it, and then I put the rest of the information in after we talk about cranial and spinal nerves. Like I said, I did a lot of rearranging with these lectures. Okay, so sensory receptors are defined as the specialized nerve endings of sensory neurons that respond to changes in environment. So this is how we know what's going on. So a stimulus is any environmental change that a receptor can respond to. Typically, receptors only respond to one stimulus. So we have a lot of different types of receptors in order to respond to different types of stimulus. The response that a nerve ending is going to have to a stimulus is going to be a graded potential. So we're going to have some sort of electrical change. When we talk about actually having sensations, that's a central nervous system thing, as is perception. So sensation is awareness of the stimulus. So unless you are consciously aware that something is happening to you, it's not a sensation, it's just sensory input. And then perception is when you interpret the meaning of the stimulus. So what that means is you might feel something, let's say on your arm, and then perception is when you realize that, you know, you walk through a spider web or something like that. So there are different levels of it. All right. So we're not going to be dealing with this part right now. We're just going to be focusing on the actual receptors. Of course, as with anything else, we have different ways to classify receptors. And in the case of these guys, there's actually 3 different things we do. And like a lot of other classification schemes, all receptors are going to be able to be classified using all of the schemes. So one thing we can use is the type of stimulus that they respond to. Another one is their location in the body. And the third one is the level of structural complexity. So this is basically their appearance or how they're made up, okay? And so all of them can fit into one of the categories in each of these. So we'll talk about that. Technically, we can also classify them based on the stimulus transduction, which is the way that they convert the stimulus into electrical activity, but we're not going to worry about that too much. So stimulus type is literally just the thing that can stimulate that particular type of receptor. And we break it up into these categories. So the biggest category, the one that covers the most things, is the mechanoreceptors. So any mechanical force, touch, pressure, stretch, vibration, any of those things qualifies a sensory receptor as a mechanoreceptor. So there's a lot of different types, and we'll look at that more later. There are also thermoreceptors, so they respond to temperature changes. Photoreceptors, this is pretty much exclusively in us, at least in our eyes. This is how we respond to changes in light level. Chemoreceptors are also a really big category, so this is responding to any chemicals in solution. And as we'll talk about later when we get to special senses, this is how taste works. This is how smell works. And then this is how a lot of things monitoring our internal environment work too. So we can monitor pH, carbon dioxide levels, lots of different chemical things in our blood and other fluids. And then the last category is nociceptors. So nociceptors respond to stimuli that could be potentially damaging to the body, and they register as pain. And nociceptors are a great example of the difference between sensory input and sensation and perception, because we talk about nociception as detecting painful stimuli, but when you're actually perceiving it, when you actually have the sensation of pain, that means you've actually processed it higher up in your brain, as opposed to just your sensory receptors picking it up. So lots of different things can trigger nociceptors. We obviously have different kinds. So that includes temperature extremes, harmful chemicals, or sometimes chemicals that we just think are harmful and hurt anyways, excessive pressure, and then of course, a lot of things that damage our tissues result in the feeling of pain as well, usually because we release chemicals ourselves that are perceived as painful. So nociceptors are the other category. These are just a couple of examples of mechanoreceptors, and these two are the ones that you should be looking at on our histology in lab. So make sure that you've done that if you haven't yet. And they do fall into another category too that we'll talk about in a minute. So when we talk about location, it's a little bit weird. So basically, we're talking about the generalized location of the stimuli. So stimuli can be external, so from the external environment. They can be internal, as in the things that, you know, we're sensing within our own bodies. And then we separate out one type of internal as proprioceptors as well. So exteroceptors are ones that are sensitive to the external stimuli that we can, wow, that's some great, whatever, stimuli. So these guys are always going to be located near or at the body surface. So most of these guys are in or under the skin, although some of them can be in other places that are close by. Interoceptors are typically monitoring our internal organs. So we also, I don't think anybody actually uses this, but you'll see them called visceroreceptors, which I think is a little more easy to remember, and that you can remember what they do. So this is all our internal organs, blood vessels, and most of this stuff we're never really aware of because this is our visceral input. And so then the third category is technically also internal, but they're specific. So proprioceptors refer to all the internal not awareness, but all the internal monitoring related to body movement and body positioning. A lot of these are also mechanoreceptors. And so they are located in and around muscles and tendons, ligaments and joints, and then other connective tissues relating to those structures. So they inform the brain and the spinal cord as to the body's location in space, the body's movements, and other things, and are then used to help coordinate and modify our muscle responses. So that's why we keep them separate from interoceptors, because interoceptors are more about like your blood pressure and your blood composition and like what's going on in your intestines and stuff like that. So then the third category is by structure. And basically we have two types. So we can just have the dendritic endings of the sensory neuron sticking into an area ready to respond to a particular stimulus, in which case we say they are non-encapsulated. There's nothing special about them. We can't tell what they do just by looking at them. And then we have encapsulated ones. And with encapsulated sensory endings, the endings have been modified in like a visible way under the microscope, or there are additional cells that are supporting the dendritic ending to make it better at detecting the particular stimuli. And I'll show you some picture examples on the next slide. Now with special senses, it's a little bit different. So special senses have sense organs. So they have entire structures that are specifically organized to help us detect those special sensory things. And we'll talk about that when we get to special senses. So don't worry about eyes, ears, taste, smell, or vestibular sense yet. We're just looking at the general senses. So these are some examples of our structural classifications. So the non-encapsulated guys, they're just, we call them free nerve endings. It's literally just the dendrites of the cell sticking into an area. We also have encapsulated guys. So then we have extra stuff surrounding the dendrite. A lot of the mechanoreceptors are like this. And then this is our specialized, and we're not going to worry about it yet. So here's a different one showing you the different categories, but here's another connective tissue encapsulated nerve ending. So again, we have all this extra structure. And you can see that both for the non-encapsulated and the encapsulated, these guys are typically those unipolar neurons with their cell bodies in those dorsal root ganglia. So all of that kind of goes together. And then when we look at the more like specialized, these are special senses, we'll look at them later, we have more bipolar cells. Oh, I've got more pictures, right? Yeah, there it is. Okay. Here's some more examples of our encapsulated versus non-encapsulated. So here's that Pacinian corpuscle that you guys should be looking at in lab. This is an encapsulated, and this is Meissner's, also encapsulated. There are other types of corpuscle-y type things as well, although we don't look at them in lab typically. And then these are the free nerve endings, and these are all the things in between. Like, they're not just free nerve endings, but they don't have as much connective tissue support. You don't need to know any of these specifically, okay? These are just examples to help you see what we're talking about. Okay, so like I said, the sensory modality stuff is just how the information is encoded, and we're not going to worry about it too much, but generally we have differences between chemical and mechanical modalities, and then photoreception's its own thing. And one more touch on the whole general versus special senses thing, in case I haven't made it clear yet. So when we talk about general senses, we mean any sensory receptors that are widely distributed throughout the whole body, whether it's internal or external or whatever. But the special senses not only have a specific organ devoted to them, but they're very focal, right? And actually all of our special senses are located on our heads. So if there's something you can only detect with your head, it's a special sense, if that makes sense. Okay, so we're going to look at how that sensory information makes it back up to the central nervous system. So we call this central processing. And again, we'll go into a little bit more when we look at somatic versus autonomic. And we're not going to get into like super detail here. I just want to give you an overview so you understand how this works. There's more detail if you want to read the book, but you don't need to know it if it's not on the slides. All right, so when we're sending information between the central nervous system and the peripheral nervous system, we're using what we call spinal tracts. So spinal tracts are basically like how there's different freeways that'll basically get you to the same place, but by different routes. And they're always multi-neuron pathways, which means we're starting with the sensory neuron, and it's going to synapse with several other neurons on its way up to the cerebrum, because that's generally where these guys end. All of the spinal tracts have 4 characteristics that they have in common. One of them is called decussation, and decussation is the fact that they always cross from one side of the body to the other. So if you remember, I mentioned in the brain that like the left side of your brain controls the right side of your body, and the right side of your brain controls the left side of your body. Well, in order to do that, all the sensory information from the left side has to cross over at some point to get to the right side of your brain. And all the motor information out from the right side of your brain has to cross over to get to the left side of your body. And so the process of crossing over is called decussation. So this is how your left brain controls your right body. And vice versa. Okay? One sec. Just mute. All right, so they all pretty much all do that for the reason I just explained. Another thing, because these are, like I said, multi-neuron pathways, another thing they have in common is called relay. So relay is the fact that we're synapsing between neurons and passing that information from one to another. So literally like a relay race where one person runs, passes a baton to somebody else, the neurons are doing the same. So we're having this multi-neuron pathway. Another one is somatotopy. So remember I showed you the weird little homunculus thing where all the sensory information and motor information in your cerebral cortex is actually organized like a map of your body. So somatotopy is, because we already know it's there in the brain, it's part of all the neural pathways. They are obviously going to different areas that correspond to location. And then the last bit is that they typically have symmetry in that the information from both sides of the body usually match. So then that makes sense too, right? So you kind of knew all this already, but we're laying it out and saying these are actually consistent things in how we get information to and from the brain. Now, when we look at the sensory pathways, we find that they're pretty much always 3 neurons. And because we have 3 neurons, here's our little bipolar or pseudo-unipolar guy. Oh boy. Hold on. Well, here. Good enough. Because it's always 3, that means that we also always have 2 synapses, right? Because the number of synapses is always going to be 1 less than the number of neurons, okay? So sensory pathways are 3 neurons and 2 synapses. And so we number the neurons based on their general location because they're always pretty much the same. So the first order neuron is that unipolar sensory neuron whose receptors are in the peripheral nervous system and cell bodies are in the dorsal root ganglion. So this is a neuron that, other than the end of its axon entering into the spinal cord, is pretty much purely peripheral. It's then going to synapse in the spinal cord with its second order neuron, whose body is in the dorsal horn of the spinal cord, or depending on where this is coming from, or it's in the medulla. So like we see that with a lot of cranial nerve stuff, sensory stuff in your face. Right, so remember we talked about how the gray matter in the spinal cord is organized in that butterfly, right? And the sensory stuff is always dorsal and the motor stuff is always ventral. So that cell body is going to be in here and that unipolar guy is going to synapse with it. And then the axon for this second order one is going to go up to the brain or in the case of the medulla, higher in the brain. And that's going to be a synapse with our third order neurons whose cell bodies are in the thalamus. So let's take a look at that with a picture. Okay, so we have sensory reception out here somewhere in the peripheral nervous system. Sensory information is running through this first order neuron whose cell body is in the dorsal root. And then we're going to synapse in the dorsal gray matter of the spinal cord. At some point we decussate, right? So at some point we cross over, and usually this happens in the spinal cord. And then we go up, synapse with the cell body in the thalamus, and then the thalamus redirects to the appropriate location in the cerebral cortex, which is where we get that somatotopy. So this one is showing you where we're synapsing in the spinal cord. This one is showing a synapse in the medulla, crossover or decussation in the medulla, and then the rest of it is still the same. All right, so we call them the ascending pathways or the sensory spinal tracts. They have a bunch of different names. And the different ones do have different names, but we're not going to worry about them, okay? That's like a level above what we're learning. So you'll see like down here, it says spinothalamic tract. This one says dorsal column system. I do not expect you to know that, but if you take a higher level neurology or anatomy and physiology, you may have to learn them at some point. Any questions about the ascending pathways? The descending are easier. All right. So we talk about the descending pathways. Now we're sending information from the central nervous system back out through the peripheral nervous system to our effectors. And it's a kind of a shorter pathway here. So when we talk about our basic motor pathways, we have a 2-neuron, 1 synapse setup. So the upper motor neuron is the first guy, and it's going to be located somewhere in the brain. So either in the motor cortex, so that's the cortical gray matter of the cerebrum, or subcortical motor nuclei, so somewhere lower in the brain. That's where the cell bodies are, and then they're going to send their axons down the spinal cord and synapse in the ventral horn. So remember, that's the gray matter with all the motor neurons in it, and the spinal cord, and then sends its axon out through a peripheral nervous system nerve and synapses with the effectors. Now this is the pathways that are actually just for skeletal muscle. It's a little bit different when it comes to like smooth muscle and glands, and we'll talk about that when we get to the autonomic nervous system. But this is how we end up with those neuromuscular junctions that we learned about before. Okay, so it's a little bit of a shorter pathway, not quite as complex, because all the complexity is occurring in the integrative part in your brain. So here's the diagram of that. So somewhere up here, we have an upper motor neuron. And we're still crossing over at some point. Okay, so we're still having decussation. And then we're going to synapse right here, and then out we go to the skeletal muscles. And remember that all the information entering the spinal cord is through the ventral root, and all the motor information, oh God, I said it wrong, that's great, is through the dorsal root, and then all the motor information exits via the ventral root, and then the nerve is here, right? Okay, so that's the descending. It's just two nerves or two neurons, one synapse, but it's still going to share those characteristics that we see, which is the decussation and the relay and the symmetry and the somatotopy, which is up here. Anybody have any questions? All right, so now that we've kind of talked about a general overview of that, like I said, we're gonna kind of go through that stuff again when we get to the somatic and autonomic nervous systems. The next thing I want to focus on is more of the anatomy. So looking at the actual nerves of the peripheral nervous system. So we divide those up into spinal nerves and cranial nerves. The spinal nerves are coming off of each segment of the spinal cord. There are actually 31 pairs, and they provide innervation to the whole body except for the head and parts of the neck. And it's really just like the front of your head and parts of your neck. As I said, they're all mixed, so they all carry sensory information back to the central nervous system and motor information back out to effectors. We name them by where they emerge from the spinal cord. So they have very similar names to the vertebrae. The difference is that there's one more cervical nerve than there are cervical vertebrae. So otherwise, though, the counts are the same as the vertebrae itself. So you know how there's 12 thoracic vertebrae? Well, there's 12 pairs of thoracic nerves. And then there's 5 pairs of lumbar. The sacrum starts as 5 sacral vertebrae that fuse together. So there's 5 pairs of sacral nerves. And then we have a single little pair of coccygeal nerves because we don't have tails. So we don't really have a lot going on down there. Now, the reason we have 8 cervical nerves is because when we start having spinal nerves coming out, they come out between the intervertebral foramina. So remember, those are the holes formed as 2 vertebrae come together, superior to the vertebra. So what that means is that above the atlas is the C1 spinal nerves. And then we go 2 through 7. But then when we get to the intervertebral foramina between the 7th cervical vertebra and the 1st thoracic vertebra, we have the 8th cervical spinal nerves. So they come out below or inferior, and then all subsequent spinal nerves, so the 12 thoracic, 5 lumbar, 5 sacral, all come out inferior to their vertebral segments, and then the little coccygeal guy comes out here. So even though we would technically have like 30 vertebrae, we have 31 spinal nerves, pre-fusion, of course. So now we're going to get into the details of the stuff that I've kind of mentioned before. So spinal nerves are formed from a dorsal and a ventral root, right? So we have the spinal cord itself, butterfly, and so we have all of these dorsal rootlets and all of these ventral rootlets, dorsal root ganglion. They all come together, and voila, you have a spinal nerve, okay? So all the motor stuff is down here on the ventral roots. All the sensory stuff is being carried by the dorsal. So all of that organization that we see within the spinal cord itself follows through this, the roots of the spinal nerve. Now, the funny thing about spinal nerves is that they branch like immediately after forming. So here's some diagrams of the roots. So again, these are the ventral roots carrying motor output. And then this is dorsal root carrying sensory input. And then this is the actual spinal nerve. But you can see that the spinal nerve branches like right after it comes out of the intervertebral foramen. Okay, can we see that? Roots, nerve, branches. Okay. All right, so the branches are called rami. Rami is the plural, ramus is the singular. And all spinal nerves branch immediately after exiting those intervertebral foramen into 4 branches. So there's always a dorsal, a ventral, a meningeal, and then there's these guys called the rami communicantes in the thoracic spinal nerves, and I will show you that in a second. These are autonomic, information only. Okay, so very diagrammatic, but here is the spinal nerve. It's like that long, and then it branches. So the dorsal ramus innervates the dorsal part of the body. The ventral ramus innervates the ventral or anterior part of the body, as well as all of the limbs. This doesn't show the meningeal branch, but the meningeal branch turns right back around, goes back into the foramen, and innervates all the meninges surrounding the spinal cord. And then these guys are the rami communicantes of the autonomic stuff that we'll talk about later. So the dorsal rami supply the somatic aspects. So this is skeletal muscle and stuff of the posterior body trunk. The ventral rami supply the rest of the trunk, so anterior and lateral, and the entirety of the limbs, so all aspects of the limbs. Now the ventral rami are a little bit more complicated than just, oh sure, there's nerves going places, because they actually come together into discrete networks that help distribute all of the axons to the appropriate nerves going to the limbs. So we have these nerve plexuses formed by the ventral rami that further complicate the spinal nerves when we look at the anatomy. So it's the ventral rami that make things complicated. The dorsal is easy. You just cover the posterior body trunk. We'll look at it again a little bit later. And then the meningeal branches, like I said, they turn right back around and they supply the meninges as well as the blood vessels in the vertebral canal, which I think is really interesting because that means like literally even inside the vertebral canal, everything outside of the spinal cord is peripheral. That might just be me though. Okay, so a plexus is when a bunch of stuff comes together. And we have 4 different plexuses, and the ventral rami of all spinal nerves except for the thoracic spinal nerves 2 through 12. So that's all the cervical, the first thoracic, all the lumbar, all the sacral, as well as our little coccygeals, all come together to form plexuses. And you can think about a plexus as the most complicated freeway interchange ever, okay? Now again, there's a lot of detail here that we're not going to cover that you may have to learn later if you take more complex classes about this stuff. So you don't need to know this. But in order to understand how and why this happens, it's handy to show. So there's different parts to it and they all have different names, but basically these are the ventral rami. And remember, there's tons of axons in each one, okay? The key to remember is that there's tons of axons. So they start crisscrossing bundles of axons and they do a bunch of it, and then when we come out of the plexus, we have a bunch of nerves supplying different parts of the body, and now all the axons are going the right place. So it's about reorganizing the axons. So like I said, there's 4 of them, and they deal with different sections of the body. So the cervical plexus is where all of the cervical vertebral rami, or at least like C1 through C5, are reorganizing to best supply the neck and the back of the head. So the cranial nerves do up here, we'll talk about those next, and then the back of the head and most of the neck are supplied by the cervical ventral rami. Well, not the very back, sorry, the back of the head. Anyway, so it's really just like those muscles and stuff that you guys had to learn recently in like the back of your scalp. Really the only one that I think is important enough to mention is the phrenic nerve. So the phrenic nerve is the only one that doesn't stay in this area and just innervate the skin and muscles of the neck and the back of the head. The phrenic nerve actually runs all the way through the thorax and controls the diaphragm, which is the skeletal muscle that's primarily in charge of breathing. So the phrenic nerve is the one that gets irritated when you have hiccups, and it's the one that's active every single time you breathe for any reason whatsoever. So it's kind of important. And it's just weird that it comes out there. Why? I don't know. The way we developed. The brachial plexus is the one that supplies the upper limbs. So these are all the nerves supplying your arms. So it's the last cervical vertebrae and the first thoracic. They have a plexus right here. It like literally runs, see how it runs between your clavicle and your ribs, and then it comes out in your armpit. If we did dissection, we'd get to see that on the cat, but we'll have to see it on our cadaver, I guess, instead. So then we have, and you don't need to know them for lecture here, well, I guess for lab if I didn't put it on there already, just whatever. I might have to add it. Probably not. Anyway, so then there's 5 of them and they deal with different things. So there's this guy, and then there's a bunch running down your arm. Your ulnar nerve is the one that runs right by your olecranon process, and when you bang it, that's what we call the funny bone. Call it the funny bone, but it's a nerve. So everything in your arm is sensory input from your arm and motor output to your arm, all supplied by this brachial plexus. Okay. The lumbosacral plexus is a combination of the lumbar plexus and the sacral plexus. So basically, both your lower limbs, parts of your abdomen, pelvis, as well as your buttocks, all supplied by these lumbar ventral rami and your sacral ventral rami. So technically there's 2 different ones, but there's a little bit of crossover between some of the branches, and so they're often referred to together. Again, you don't need to know any of that, okay? It's just because I think pictures are important. And I found so many cool pictures just to kind of show you how it all comes out. So this is just kind of showing you them all coming out. Plexus-y up here, plexus-y down here. We have a variety of very important ones, but you can see that then there's a lot of stuff emerging down here, including that sacral nerve or the sciatic nerve that a lot of people get irritated because it's very, very big and it runs right there. Okay, so so that's that. That's this whole area, lumbosacral plexus, including your little coccygeal. They don't do much. They cover like a teeny tiny area by your anus. Okay, so There are a couple of ventral rami that don't form plexuses though, and those are your thoracic spinal nerve ventral rami. So instead, they form what are called intercostal nerves. So every ventral ramus just runs between two ribs and innervates the anterolateral thorax and abdominal wall. And because they're emerging from one segment to another, it's a simple segmental pattern. So they supply like your intercostal muscles, the muscles of the skin and anterolateral thorax, and most of the abdominal wall, and as well as the skin and stuff. So they're intercostal because they're all running in between ribs, right? Between ribs. And this is T2, T11. And then the ventral ramus T12 spinal nerve is technically called the subcostal rib because it emerges inferior to the 12th rib. And so there's no other rib in between it. Poor thing, has a different name. Not a big deal. Probably not anything I'm going to mention ever again, but just showing you how we try to name things descriptively. So when we look at that, we again see there's our dorsal ramus, there's our ventral, and it just runs very straightforwardly, branching superficially and deep to supply sensory feedback back to the central nervous system and then muscle output to all of those skeletal muscles. Now we see that the dorsal rami of all spinal nerves do the same thing. So just like these thoracic intercostals have this segmental pattern because there's no plexuses, the dorsal rami all do the same thing. So we have a segmental pattern of innervation all up and down our backs. So along the whole posterior aspect of our trunk, each dorsal ramus innervates a narrow strip of muscle and skin. So basically, I have a picture later, but like really it looks like this, okay? And so the fact that it innervates skin, so it's carrying sensory information from the skin, means that we have these strips of skin that are being supplied by a single nerve. And so we call these sections dermatomes. And the significance of a dermatome is that if your spinal nerve was damaged or like your spinal cord was damaged at a certain level, then you might find areas of your skin going numb because now we've disrupted the information being carried from your skin back to your central nervous system. Now there are branches, so there's a different branch of each of these nerves supplying the skin that are called cutaneous branches, and then there's deep branches to the muscles. And we find that even with the ventral rami, there are little strips of skin on the front of our bodies and our limbs that correspond to a single spinal nerve. And so if we're trying to determine which nerves are damaged, if somebody has a spinal cord injury, we're trying to localize the area of injury, we can actually test for sensation along the body to figure out which ones are damaged. So we end up with this body map showing you which spinal nerves are innervated at which levels. And so you can see they color-coded it to show you which one's which. And so even things like your second cervical segment provides or carries sensation for like the whole back of your head. So if the back of your head went numb, you probably have a lot of other things to worry about, but you know, they would be looking up here, right? So yeah, it's a whole map basically of innervation by spinal nerves. Okay, so that's what dermatomes are. Kind of cool. I don't know how much they actually use them in practice anymore because, you know, you can put somebody in an MRI or a CT and see where the damage is, but at least before we had a lot of imaging, this was very useful. All right, so unless somebody has spinal nerve questions, I'm gonna move on to the cranial nerves. Okay, perfect. So the cranial nerves are to the brainstem as the spinal nerves are to spinal cord. Now there are 12 pairs of nerves associated with the brainstem, or the brain, technically. Some of them are not the brainstem. And they're in charge of sensory input and motor output to the head, and then a little bit of the neck, the rest of the neck that isn't covered by the cervical. Because we don't have spinal cord segments, instead they got named by structure or function, which you'll see as we go through. And they were given numbers by Roman numeral, which is stupid, but it's the way it is. From rostral to caudal. So basically, I'll show you when you look at the picture. But basically, you know, they were like, okay, the ones that are closest to the front or most anterior when we look at the brain are number 1, and we go back from there. Now, just like spinal nerves, most of these guys are mixed, so they carry both sensory and motor information, but there are some that pretty much just carry sensory or just carry motor information, although when you really parse it down to the details, even that's not completely true. So I don't need you to know that, but sometimes you have to memorize that for different classes, and there are mnemonics for that, but you will not need to know that for my class. I don't care about it because I don't think it's important, to be honest. So here's an inferior view of the brain, and all of the spinal nerves are on here. And so these are generally what we use to study this stuff. If we were still meeting in person, you know, for lab, we would have some brain models with this flipped over, and instead we'll have those cadaver pictures with the brain, and you have those cranial nerve options to help you study. So we're going to go through these guys and in general what they do, and then how people usually study for this. So cranial nerve number 1, we'll talk about a little bit more when we get to smell, because this is the olfactory nerve. This is just carrying the fibers with sensory information from smell into the brain. So the weird thing about the cranial nerve number 1 is that it's not really what we identify isn't actually the nerve itself, because the nerve itself is really, really short. It's just running right through the ethmoid bone, and then it immediately synapses in what we call the olfactory bulbs. So we never actually see it, because once you take a brain out of the skull, you've ripped those nerve fibers, and it's not like a clump. It's through a bunch of different little holes. So when we say olfactory nerve, really what we're identifying is the olfactory bulbs, which is actually the location of our second order neurons. We just ignore that for purposes of cranial nerves, because that just is extra information. So I'll go into a little bit more of that when we actually get to smell, but that's olfactory or cranial nerve number 1. Cranial nerve number 2 is the optic. So the optic nerve carries all the visual information from your eyeballs. So again, we'll talk about it a little bit more when we get to special senses. So basically, the optic nerve comes off of the back of your eyeball. It goes through the optic canal into your skull. And then it does this fun thing where half the fibers cross over and go to the other side, and half of them continue on to the same side. And we'll talk about that later. But it forms a fun thing called the optic chiasm. And then after that, it's the optic tract, because now it's considered to be part of the central nervous system. So remember, nerve is white matter in the peripheral, tract is white matter in the central. And again, we'll talk about that more when we get to special senses. So those two are pretty straightforward. Obviously, it's going to get more complicated from there, because it can't all be that easy. So before I tell you about the third one, I need to tell you a little bit about the muscles that let us move our eyeballs. So they're called extrinsic eye muscles. And There are 6 of them, and there are 4 rectus muscles because they have straight moving stuff, and they let us do things like move the eyeball up, down, left, and right. And then there are 2 oblique muscles, and they give us the full range of motion to let us move our eyes however we want to, if that makes sense, in combination. So there's an inferior oblique here, and there's a superior oblique up here, okay? So we don't have time to go into the nitty-gritty details about this, but basically, it's one of the other things taking up all the space in your bony orbit other than your eyeball. Oh, and here's your optic nerve right here. Cranial nerve number 2. Okay, so there's actually 3 different cranial nerves controlling these extrinsic eye muscles, and they control different ones, and so therefore you kind of have to touch on, we have 6 eye muscles that let us move our eyeballs, okay? So that is why I bring that up right now. So cranial nerve number 3 is the oculomotor. It controls most of the extrinsic eye muscles, and then it has sensory information about what those muscles are doing that it sends back. So proprioceptive afferents. That makes sense. It also carries some autonomic stuff, so parasympathetic fibers to the pupillary, sphincter, and ciliary muscles. And we'll learn more about that later, but basically this is smooth muscle inside the eye, okay? So some autonomic stuff's hitching a ride here, and we'll talk about that more later on. Number 4 is the trochlear muscle, and the trochlear muscle controls a single extrinsic eye muscle, the superior oblique. Now, the reason it's called a trochlear is because this oblique muscle, in order to insert at an angle on the eyeball, runs through this little connective tissue pulley, and trochlea means pulley. So we named it after that, and that's what it does, okay? So 1 muscle controlled by this nerve. Now 5 is super important. It's called the trigeminal because there are 3 major branches off of it, and it's sending pretty much all of the sensory information from your face back to your brain. It also carries the motor efferents for your chewing muscles. So that's pretty much like your masseter and your temporalis, and then some little guys in there that I didn't make you learn. And you can see the divisions are just kind of dividing up the face. So the ophthalmic kind of does the eye and everything above that. The maxillary kind of covers where your maxillary bone is, and then the mandibular does all that stuff. And you don't need to know the divisions, showing you what it is. So again, this is number 5. And then I put this picture in because it really shows you how we have these 3 big branches, and that's what gives it its name, okay? Trigeminal. Now the 6th one, cranial nerve 6, is the last of the control of the extrinsic eye muscles. It's called abducens. It only controls the lateral rectus. So the lateral rectus is the muscle that lets you turn your eye laterally. So using the right one, and then I'm using the left one. And what do we call it when we move something away from midline? We call it abduction. So because this muscle lets us abduct the eyeball, sort of, they called it abducens. And then, as I said, the other 4 are controlled by that cranial nerve 3 oculomotor. And that is all of the eye ones. Okay, so next is 7. So 7 is facial. And 7 is basically the opposite of the trigeminal. So the trigeminal is the sensory information from your face, and then the facial is the motor control over your muscles of facial expression. So all of the muscles in your face, other than the chewing ones, are controlled by the facial nerve. And again, you don't need to know this, but there's 5 branches. So this one controlling different sections of your face. And there's also a lot of autonomic input here. So we're also controlling some involuntary effectors. So a lot of the glands, including our tear glands, the glands in our nose that would make our nose run, and most of our salivary glands, all controlled through this nerve as well. And it also is the sensory impulses from the anterior 2/3 of the tongue. So most of what we taste with is also carried by this nerve. So this nerve does a lot. It lets us control all of our face. It controls most of our salivation as well as other glandular secretions. And it carries a lot of taste taste information as well. So this one and the trigeminal are kind of big for facial stuff. Again, you don't need to know this right now, but I just find it amusing that they come up with a mnemonic using somebody's hand. I don't know how that's meant to help, but there you go. All right, so then number 8. Number 8 we're going to talk about more when we get to special senses, because number 8 is the vestibulocochlear nerve. And it carries all the sensory information from both your hearing as well as your equilibrium special senses. So your equilibrium is also called your vestibular sense, which gives us the first part. And the sense organ for hearing is the cochlea, so vestibulocochlear for equilibrium and hearing. There are a little bit of motor stuff in there, but we don't really worry about it too much. And so these guys come in from a bunch of temporal bone, and we'll talk about that when we get to those senses. All right, now things get slightly weirder. So glossopharyngeal is cranial. Did I do that again? Oh my God, I never fixed that. Okay, sorry. This is 9, guys, not 4. So this is cranial nerve number 9. And it innervates the tongue and the throat. And that's where its name comes from. So glossa refers to your tongue, and your pharynx is the muscular part of the throat that's right behind your nose and your mouth. So glossopharyngeal because it's tongue and throat. So your throat muscles are mostly involved in swallowing. Any other glandular stuff is controlled here. So this is the other salivary gland gets controlled here. The taste to the back of the taste from the back of your tongue is carried by this nerve, as well as sensations to your mouth and your throat. And this guy also monitors the carotid body and the carotid sinus, which are part of how we monitor blood pressure. Talk about that more when we get to autonomic. So this guy does a lot of stuff, but it's mostly in here, mouth, throat area, okay? So we're kind of moving, we've moved away from the face itself, and we're kind of going internal and back, okay? There is like a general map to this. All right, now that takes us to arguably the most important cranial nerve we have, which is cranial nerve 10, or the vagus nerve. Now, the vagus nerve is the only cranial nerve that innervates anything beyond the head and the neck, which is what makes it the most important. So it does do a little bit of stuff in the head and the neck. It innervates some of the other muscles in the throat for swallowing, so it works with glossopharyngeal. But the important thing is that it carries a lot of visceral sensory information from the thorax and the abdomen back to the brain. and it sends the visceral motor output to the thoracic and abdominal viscera as well, okay? So this guy is kind of a big deal. It carries a lot of autonomic output and input. And we're kind of still learning about the implications of that fact too. So here's vagus, and you can kind of see it goes through and has input in the thorax, so heart, lungs, airways, stomach, other intestines, liver, kidneys, spleen, all of that stuff has innervation from the vagus nerve, to the point where there have been studies about using nerve stimulation to the vagus nerve to help treat conditions, especially of thoracic and abdominal viscera, which we do not have time to go into, but here's some references if you're curious. All right, so anticlimactic after vagus, but 11 is the accessory nerve. Now the accessory nerve just controls the trapezius and sternocleidomastoid muscles. So hopefully you remember sternocleidomastoid, they're these guys that run right here. And of course the trapezius is that big guy on your back. So this is why the cervical plexus doesn't control everything on your neck, because the accessory nerve controls these muscles. Don't know why. Because it controls the muscles of the neck, it used to be called the spinal accessory or accessory spinal nerve, but they decided that spinal made it confusing because it's a cranial nerve, so now it's just the accessory nerve. So then finally, we have 12. So cranial nerve 12 is the hypoglossal. So remember, glossa refers to the tongue, hypo means under. So this is what controls all the muscles that actually control the tongue. So it's motor for tongue control, which means that when we're like chewing and swallowing food and your tongue is doing things, it controls that. And also the way this is that we move our tongue when we're talking. So it's, yeah, so tongue muscles. No, that's it. All right, so hold on, do I have one? All right, so yeah, learning their cranial nerves is kind of a mandatory, gotta say, so kind of have to throw it in there. And there's lots and lots and lots of mnemonics out there to help you. So you're gonna focus on knowing, for lab, you just need to know their names, both the number and the name, and then their location on the brain, right? So for lecture, what you kind of want to just concentrate on is their general functions. You don't need to know every single detail, but you just need to know the general functions. Like I said, there's some people care about motor and sensory, and I don't care. But I throw them in case you need it later. And this is something that's, I don't even remember anymore, but it's something related to this. So like I said, there's tons of mnemonics. I always like to throw a bunch on there. And don't worry about the motor, but some of them are just stupid, some of them are funny, some of them are dirty, whatever helps you remember. And I do encourage you to make up your own if that works better for you, because if you make up your own, you're going to remember it better. But if there's one that you really like, then go for it. Okay, there's tons on there. There's more than I have found, I'm sure. These are just some of the either classic ones or funniest ones that I found. This is a really weird one where it's a visual mnemonic to help you kind of remember what they do based on where they write the numbers. It doesn't make any sense to my brain, but if your brain works like this, then more power to you. There's even Harry Potter ones, so I'm sure there's nerd ones for anybody that's into a specific genre or thing. Lots and lots and lots of mnemonics to help you learn this stuff. okay? Yeah. And then another way to help learn it, you don't have to do these, but if this is how you learn it too, is these are some links to how to actually examine or assess cranial nerves, which usually we do in lab, and I don't think we're actually going to get there this time because I still haven't seen a good simulation for it. So we'll just concentrate on the anatomy, but if it helps you to do it, to understand, then you can kind of look through how it's tested, and that should help you remember what they do. So for example, we test the olfactory or cranial nerve 1 by literally seeing if you can smell things. Which, you know, that one's not bad. So again, the reason why they're numbered 1 through 12 is because of how they emerge from the brain. So because olfactory is here, it's first, and then the optic nerves come out of these optic chiasms here, so they're number 2. And then the chiasm is where it crosses over. So then oculomotor are these little guys here, that's number 3. And then number 4 is trochlear. And then you'd think, like, right, like, doesn't it make sense to just keep the abducens with them so that we're not splitting up our eye control? It would, but here's the trigeminals. So they're next. So they have to be number 5. So then abducens isn't until number 6 because it's next, okay? And then here's our big old facial nerve here. Here's our vestibulocochlear. And then glossopharyngeal, 9. Vagus nerve is 10. This is where things get a little bit weird. So here's our accessory. This is the other reason why they used to call it the accessory spinal, because it's like literally running down the beginning of the spine. And then here's hypoglossal. Okay, so there's always going to be a right and left. They should always be in exactly the same place. Some of the diagrams are different from others, so make sure that you're studying the actual brain thing on the McGraw-Hill Connect, because that's what I'll use for your test, because that's what I expect you to be studying off of. Because I've had people tell me like, oh, well, I studied this exact diagram and it really threw me off because it looked a little different. And you're like, well, they're all trying their best to replicate reality, and what we have is a picture of reality. So study the reality one and not the diagrams. Anybody have any questions? Okay, so we'll do the rest of the peripheral nervous system on Wednesday, and then I'll do an extra lecture to get us through the special senses. Since we're not quite close to special senses yet, I don't really want to launch the lab stuff until Wednesday as well. So we'll do the special senses pre-lab then. So just stick with what you already have for lab for the moment, okay? And then I'll add on Wednesday. Any other questions?